Hi again, and welcome back to our final video on the Great Oxidation Event, which is on the biological consequences of the increase in oxygen, which opened opportunities for new groups to evolve. So I won't talk about this much because you've heard about the timing before. So after the Great Oxidation Event, oxygen levels were still much lower than they are today, but even then, 1 to 10% of today's level, so maybe about 1% of the atmosphere or so, was still enough to open up new evolutionary opportunities. Now, there's some hints for a temporary decrease in oxygen about 1.9 billion years ago. That'll become important potentially towards the end of this video. But first, what consequences did the Great Oxidation Event have for life? Well, prior to the GOE, life would have been bacteria and archaea that didn't need oxygen. But there is a third group of life, the eukaryotes. And so eukaryota includes all of the large multicellular organisms that you're familiar with. Animals, fungi, plants are all eukaryotes. Eukaryota also does include many single-celled organisms belonging to a wide range of different groups, but collectively referred to as protists. Because virtually all eukaryotes, whether they're protists or animals or plants, require some oxygen, evolution of the group has long thought to be linked with the Great Oxidation Event. So as I mentioned very briefly in an earlier video, the evolution of eukaryotes is really a fascinating story. So basically, an archaea cell brought a different bacteria inside of it, ultimately forming a partnership, also called a symbiosis, as the two merge to become a different type of cell, to become a eukaryote. Our cells actually still contain these ancient bacteria, the mitochondria, that play a major role in our metabolism. So when this hypothesis was first proposed by Lynn Margulis in the early 1970s, it was an extremely controversial idea, but there's now a tremendous amount of evidence supporting it. And believe it or not, this wasn't the only episode of endosymbiosis. It also happened again in a separate event when a eukaryote cell merged with a cyanobacteria, which gave some eukaryotes, including modern plants, the ability to perform photosynthesis. So what evidence do we have for eukaryote evolution to know when they evolved? Well, first we have to figure out how to tell whether something is a eukaryote or not. And one possible clue could be size. Because eukaryotes tend to have larger cells, usually 10 to 100 times as big as a bacterial cell. And this is because eukaryotes can actively transport material around within the cell. In contrast, bacteria rely on a process called diffusion, which is kind of the passive spreading of material, and that tends to limit their size. So there are some big bacterial cells and also some really big eukaryote cells, but things that are between 10 and 100 microns, which is millionths of a meter, so 100 microns is about the width of your hair. So cells about that size are much more likely to be eukaryotes than they are to be bacteria. Eukaryotes also have something called a cytoskeleton, which is made of tiny tubes that allow the cell to move and change shape, capture other cells, otherwise bring in outside material. And the complexity of this cytoskeleton is one thing that distinguishes eukaryotes from bacteria and archaea, so we might expect to see more complicated cell shapes in eukaryotes than we do in other organisms. And finally, eukaryotes can have true multicellularity, although many of them are single-celled as well, it's true. Now, of course, also some cyanobacteria can form colonies that have multiple cells, like in the photo here, but in this case the cells are all fairly similar. In multicellular eukaryotes, different cells can specialize for different purposes, such as the specialized reproductive cells in this red algae here. So let's look at some fossils. So here's an organism named Bangiomorpha, which lived just a little over 1 billion years ago. So let's look at our 3 possible indicators for eukaryotes and how does Bangiomorpha do. Well, first, the cells are pretty large. You can see the scale bars in some of these. They're typically 10 to 25 microns in size. That's within our normal range for eukaryote cells and much bigger than most bacterial cells are. So, check. We'll give it a check for that. There are a variety of cell shapes as well, so we'll give it a check for complex cell shape that would be consistent especially with the cytoskeleton. And finally, there are different cell types, so we'll give it a check as well. There are reproductive spores. There are cells that are specialized to be an attachment structure, for example. So what's our verdict here? Well, Bangiomorpha is widely accepted as a definite eukaryote, and in fact, as a red algae belonging to one of the groups still alive today. So because of that, it's probably not one of the earliest eukaryotes. It did also live more than 1 billion years after the Great Oxidation Event. So moving back in time, there's a group of organic fossils called acritarchs, and this probably includes many different types of organisms, but acritarchs can be found in rocks as old as 1.65 to 1.8 billion years ago. So are these eukaryotes? Well, first, do they have large cells? Well, you bet they do. Acritarchs are usually tens to even hundreds of microns in size. Although some bacteria can reach that size, it's quite rare. Well, eukaryotes that's a pretty typical size for their cells. Next, do they have complex features? Well, indeed they do. Some acritarchs, like the ones here, had spiny surfaces or cell surfaces made of plate, such as the one on the right. Pretty complex cell coverings and textures. Did they have different cell types? Well, acritarchs are single celled, so this doesn't really apply. The verdict for acritarchs is then, well, yeah, most people accept acritarchs as pretty conclusive, pretty likely eukaryotes. Finally, we'll consider a fossil called Grypania, which is a carbon filament found in rocks as old as 1.87 billion years. So what does it have? First, large size. Yeah, we'll give it a point here, because these individual filaments are several millimeters across and up to several centimeters long. Now, individual cells aren't preserved, so we can't tell cell size, but these are pretty big. Next, do they have complex cell shape? Probably not, although again we can't see the individual cells, but the filaments are pretty uniform and simple looking. Finally, do they have different cell types? Well, this is unknown, but again, there's not really any strong evidence for it, again because of the simple shape of the filament. So what's the conclusion? Well, Grypania could possibly be a eukaryote given its size. It's quite large, but we can't rule it out being, say, a bacterial colony or something else. Now, you might remember from last class that the fossil record will normally underestimate the true age of appearance of a group because we aren't likely to find the oldest one that ever lived. Perhaps it didn't fossilize, or it did but the rocks have been destroyed, or the rocks are around but we haven't found it yet. There are lots of reasons. Now, you might also remember that there are some other methods that we can use, so let's take a look at them. So you learned about biomarkers in the section on cyanobacteria evolution, and they can also be used and have been used to search for eukaryotes. It's a different biomarker. In this case, the specific molecules are called sterols, which are thought to be formed only by eukaryotes, although it does turn out that perhaps some other groups can make them rarely as well. There are a bunch of different sterols. Cholesterol is probably the best known because of its association with our health. Now, these sterols tend to break apart a little bit, so the molecules that we find in ancient rocks are called steranes, which are just sterols with some of the bits broken off. So there was a report of sterane biomarkers in rocks that are 2.7 billion years old. Now, as you might recognize, those are before the Great Oxidation Event. Now, the graph here just shows an example. So like with the other biomarkers, people look at these by finding and identifying peaks on these spectrum that separates out all the different molecules by how much they weigh. So finding evidence for eukaryotes in 2.7 billion year old rocks would be a huge discovery because it would mean that they're way older than generally thought and that they didn't need oxygen to evolve, which is also generally thought. Well, however, it was later discovered that the organic material that actually seeped into the rock much later on. So the evidence for them being 2.7 billion years old is no longer considered to be very good. So now the oldest sterane biomarkers are about 1.65 billion years old, which is about the same age as some of the likely fossils, such as acritarchs, for example. Well, like with the cyanobacteria before, perhaps we can use a molecular clock approach to estimate the origin of eukaryotes. Just as a quick recap, These methods use basically the amount of genetic differences between a bunch of living organisms to then calculate and extrapolate back to estimate when those groups first split apart from some common ancestor. The family tree here shows a whole bunch of eukaryote groups. If you look very carefully, you might see some animal names, if you know scientific names, within this group called Opisthokonta at the bottom. And the tree also shows how these groups are related to each other. So from all of this, the best estimate is that eukaryotes first evolved about 1.75 billion years ago, although that result does depend on the specific methods used. And so with different methodological choices, the estimated age could be anywhere between a billion and maybe 1.9 billion years ago. So all of these lines of evidence seem to agree on a consistent timing about 1.6 to 1.8 billion years ago. But in the last 10 years or so, a new finding has kind of shaken up this field. These come from 2.1 billion-year-old rocks in the country of Gabon in West Africa, or equatorial Western Africa. They've been called the Francevillian Biota, and they were, to be honest, a very surprising discovery because they're really big. I mean, at least for this time period. Some of them are 10 to 15 centimeters long, which might be about as long as your hand is. So even Grypania, which is pretty big, is maybe only about the size of a penny when it's coiled up. So these things are big, and that large size raised the possibility that maybe these are fossils of multicellular life. But they're also quite irregular in shape, and to be honest, a little weird looking. And that prompted other scientists to question whether they might be, say, maybe bacterial colonies, such as microbial mat pieces, or perhaps not even remains of living organisms at all. Maybe they're what are called pseudofossils, fake fossils that aren't even, that are inorganic and not evidence of life. Well, the scientists went back and did some more investigations and found some more things, and they found some features that look a lot like burrows or little trails. In some cases, with a potential fossil actually preserved at the end of one of these little trails. And like in D here. And so they interpreted this as active movement by the organisms. And that would really be pretty good evidence for some kind of multicellular life rather than a colony of bacteria, for example. Or it could at least suggest these are complex cells that are able to move around. But is this the only way we could explain these trails? Do they have to indicate some creature actively moving around on its own? Well, some experiments have actually shown that it's possible, and perhaps relatively common, for little fragments of microbial mat to get broken off and then drift or get pushed along the seafloor, making grooves as they go. So in these little experiments here, these microbial mats are made up entirely of bacteria, cyanobacteria. They're not actively moving, so there may be no relationship between grooves, even grooves with little things at the end of them, and multicellular organisms or complex cells. All right, so let's summarize what we know from all of this. So the fossil, the chemical biomarker, and the molecular clock evidence all suggest that eukaryotes evolved somewhere in the range of 1.8 to 1.65 billion years ago. Possibly a bit earlier if Grypania is a eukaryote, but it might not be. And there's good evidence for representatives of modern groups, such as red algae, green algae, and amoebozoans, like this cute little vase-shaped critter here, between about 750 million and 1 billion years ago. But then there's the Francevillian Biota about 2.1 billion years ago. So what's the deal with that? Well, on the one hand, perhaps they aren't multicellular life at all, right? Perhaps they're just bacterial colonies, or maybe some inorganic thing, right, that's not even life. So in that case, the eukaryotes might have evolved right after this potential oxygen crash about 1.9 billion years ago. Assuming it exists, there's still some, still not fully accepted potentially. But what if the Francevillian Biota really were multicellular life? Well, if that's true, it raises the possibility that multicellular life and complex cells evolved prior to 2 billion years ago, but maybe they actually went extinct if there was this oxygen crash, and then modern eukaryotes evolved separately afterwards. Now, if that happened, that might mean that complex life on Earth evolved on two separate occasions, independently, which would really be an amazing finding. Well, for now, the interpretation of this Francevillian biota is still pretty debated, so we don't know which of these hypotheses is more likely at this point. But what do you think? Did multicellular life evolve twice? Or is the Francevillian biota bacterial colonies or something else? Thanks for listening.